All right, let's continue further. So now let's look at nucleotides. So nucleotides are organic molecules consisting of a nucleoside and a phosphate. They serve as monomeric units of the nucleic acid polymers. Called deoxyribonucleic acid, in other words, DNA, and ribonucleic acid, RNA. Both of which are essential biomolecules within all life forms on Earth. Nucleotides are obtained in the diet and are also synthesized from common nutrients by the liver. Going further, so a polynucleotide molecule is a biopolymer composed of 13 or more nucleotide monomers covalently bonded in a chain. Yeah, so you have nucleotides. It consists of a nucleoside and a phosphate, and a polynucleotide is it's composed of 13 or more of these monomers covalently bonded in a chain. Let's just go illustrate this here. This is let's make this a bit smaller. So this nucleotide contains the 5-carbon sugar deoxyribose at center. So this is the 5-carbon. So 1, 2, 3, 4. And I believe this is the 5th one there. Yeah, so this nucleotide contains a 5-carbon sugar deoxyribose at center, a nucleobase called adenine. Or actually, I think it's pronounced adenine. So that's upper right here. That's a nucleobase. And one phosphate group on the left. There's a phosphate there. There's a sugar, and there's this. And that's pretty much a nucleotide is a nucleoside and a phosphate. Nucleoside would be, I believe, these two combined, and this would be the phosphate over there. So phosphoric acid, also known as orthophosphoric acid or phosphoric acid V or phosphoric V acid, is a weak acid with the chemical formula H3PO4, and then right here the pure compound is a colorless solid. So this is how it looks like right here, and these distances are from the atoms, so 152 picometers, 157 picometers. A structural formula of a phosphoric acid showing dimensions, and a picometer or PM is 1/1,000,000th of a meter, so 1 PM equals 1 times 10 to the negative 12 meters. Okay, going further, so a phosphate is an anion, so it's negative charge salt, functional group, or ester derived from a phosphoric acid. So a salt, if you recall, it's when you have a net neutral charge 0, but it's composed of the equal amounts of positive and negative ions. So yeah, so phosphate is either an anion or a negative charge salt, functional group, or ester derived from a phosphoric acid or phosphoric acid. It most commonly means orthophosphate, a derivative of orthophosphoric acid H3PO4. So a derivative of this one, it's a derivative of this, and the phosphate or orthophosphate ion PO4, and then with a charge of 3 minus there, is derived from phosphoric acid by the removal of 3 protons. So if you have this H3PO4 and you remove 3 protons, you can get PO4 and with a negative 3 charge, and that's what this phosphate usually refers to, or the phosphate ion. And the removal of 1 or 2 protons gives the dihydrogen phosphate ion H2PO4 minus and the hydrogen phosphate HPO4 2 minus respectively. So yes, here's a stereo or 3D skeletal formula of a phosphate anion or phosphate ion. This one is 3. This can be there's no H anymore, so this can be PO4 and 3 minus. This is all negative charge. Free phosphate anions in solution are called inorganic phosphate, generally denoted Pi, and at physiological or homeostatic or steady internal conditions, pH. So at physiological, just basically steady state conditions, pH. At steady pH primarily consists of a mixture of HPO4 2 minus and H2PO4 minus ions. Yes, continue further. So a nucleoside consists simply of a nucleobase, also termed a nitrogenous base, and a 5-carbon sugar, either ribose or deoxyribose. And ribose has the chemical formula C5H10O5. Deoxyribose has the chemical formula C5H10O4. So deoxy, so it's removing one of these oxygen ones. So deoxy sugars are sugars that have had a hydroxyl or a OH group replaced with a hydrogen atom. and aka loss of an oxygen atom. Yeah, so you remove the oxygen right here. So notice H5, this is H4. So it's a ribose, and then you have a ribose with a loss of an oxygen, or deoxyribose. So nucleoside consists of this and a 5-carbon sugar. So if you go back to our nucleotide initial one here, so you have, so it includes this and this. This is a nucleoside. So that's a nucleoside, and then this nitrogenous base is the nucleobase. And there's the 5-carbon sugar, the ribose or deoxyribose. In this case, it's a deoxyribose. So it's loss of 1 oxygen. Let's continue further. And yeah, so here is an image of ribose and deoxyribose. So here you have, this is including all the H's, all the hydrogens. So yeah, so there was that carbon there. So this is the way labeling, it starts from here, 1, 2, this is carbon 1, carbon 2, 3, 4, and then 5. And then this part right here, this is ribose as an OH, deoxyribose just as an H. So it's removed that oxygen atom there. So figure the difference between ribose and deoxyribose is the presence of a 2-OH. In other words, what this means is at this 2 spot. So at this 2 spot, you have an OH there. That's the difference. Is there's an OH for ribose, and it doesn't have an OH there. It just has an H at the deoxyribose. So note that the carbon atoms are labeled from 1 to 5. Just keep that in mind. So nucleobases, also known as nitrogenous bases, or often simply bases, are nitrogen-containing biological compounds that form nucleosides, which connect to a sugar, which in turn are components of nucleotides. Nucleotides with all of these, and nucleotides has that extra phosphate, with all of these monomers constituting the basic building blocks of nucleic acid. And the ability of nucleobases to form base pairs and to stack upon another leads directly to the long chain helical structures or helix structures such as ribonucleic acid RNA and deoxyribonucleic acid DNA. Yeah. So 5 nucleobases. Yes, yeah, 5 nucleobases. There's 5 of them. Adenine, adenine A, and then cytosine C, guanine guanine, and then G, and then thymine T, and then uracil U, are called primary or canonical. Yes, or canonical nucleobases. They function as the fundamental units of the genetic code, with the bases A, G, C, and T being found in DNA, while A, G, C, and U, so replace the thymine with uracil, are found in RNA. So very interesting stuff. And going further, you can further categorize these as Purines and Pyrimidines, or actually Pyrimidines. I think that's better pronunciation. So so right here, so Purine nucleobases are fused ring molecules. This is the AG, so you have 2 rings right here, and they're fused together. And then these are just the Pyrimidines are nucleobases have just simple ring molecules. So just a ring right there, ring right there, ring like that. And then the fused ones are A and G. Yeah, so A, G, and then while CT, this T is for DNA, and then uracil is RNA. CTU are single rings, simple ring molecules like that. So if you go back up to our starting point of the nucleotide, and right here, this one is a fused ring that is going to be a purine, and not a pyrimidine. So yes, very fascinating stuff. All right, so let's go further and look at let's look up the term base pair. So a base pair, or BP, is a fundamental unit of double-stranded nucleic acids consisting of 2 nucleobases bound to each other by hydrogen bonds. So yes, you get these nucleobases connected to each other. That is going to be a base pair. So, for example, this AGCTU, if they connect with hydrogen bonds, that's a base pair. They form the building blocks of the DNA double helix and contribute to the folded structure of both DNA and RNA, dictated by specific hydrogen bonding patterns. And this is called so then you have these types, the Watson-Crick base pairs, or the guanine and the cytosine pair, and adenine and thymine pair, or I'll just say GC and then AT. Allow the DNA helix to maintain a regular helical structure that is subtly dependent on its nucleotide sequences. It depends on the sequences, and that forms the code as well for genetic material, or just for building as building blocks for bigger substances such as proteins and so on. So the complementary nature of this based pair structure provides a redundant copy of the genetic information encoded within each strand of DNA. Yeah, and this complementary nature, going over, so complementarity is the base principle of DNA replication and transcription. In other words, DNA to RNA, as it is a property shared between two DNA or RNA sequences, such that when they are aligned anti-parallel or parallel but reverse directions to each other, the nucleotide bases at each position in the sequences will be complementary, much like looking in the mirror and seeing the reverse of things. In other words, you're seeing the same thing, but in the opposite direction. So they're complements of each other, the two strands. And we'll illustrate that further. So adenine and guanine, guanine, are purines, while thymine and cytosine and thymine, cytosine, and uracil are pyrimidines. Purines are larger than pyrimidines, both of which, pyrimidines because that's a simple ring structure, these are the fused one. Both types of molecules complement each other and can only base pair with an opposing type of nucleobase. Yeah, so you'll have purines connecting to pyrimidines. Let's just double check that case here. So ATG and then C, then AUGC. Yeah, so you have AT, GU, or GT, yeah. GC. Where's the G? And then there's a C, yeah, so GC, yeah. So you're going to have to mix up on the opposite, either pyrimidine or, and purine. So both types of molecules complement each other and can only base pair with opposing type of nucleobase. In nucleic acid, nucleobases are held together by hydrogen bonding, which only works efficiently between adenine or adenine and uracil. Or actually, I'll just move this over. This is better to written it over here. Let's just move this like this. Copy this, delete, and then put it here. So this hydrogen bonding nucleic acid and nucleic acid nucleobases are held together by hydrogen bonding, which only works efficiently between adenine adenine and thymine, thymine or adenine and uracil in RNA. This is for DNA, and between guanine and cytosine in DNA. The base complement A equal T, or shares two hydrogen bonds, so that's a two hydrogen bonds there. While the base pair G and then this three line equal sign C has three hydrogen bonds. All other configurations between nucleobases would hinder double helix formation. DNA strands are oriented in opposite directions. They are said to be anti-parallel. So it's the same, but in opposite directions. So here we have nucleic acid. So DNA and RNA, so nucleobases are the A, T, G, C. And then in RNA you have A, and replace the T with C. With U. Replace with U, and then G and C. And then the base complements are AT. A connects to T, and then G connects to C there. Here we have AU, and then this is GC, and these are triple hydrogen bond to GC, but then the A to T and A to U are double hydrogen bond. Yeah, and let's have a better look at these hydrogen bonds. So here's adenine and thymine, so they match up between two DNA bases, guanine and cytosine, showing hydrogen bonds, dashed lines holding them together. Whoops, I think I copied and pasted the wrong heading there. This one's over here. This one is over here. And this one is the adenine and... yeah, this one is over here. So let's copy this. So, and this one goes here. Yeah, so matching between two DNA bases, adenine and thymine, showing hydrogen bonds, and then dashed lines there, hydrogen bonds holding them together. So yes, this is the two hydrogen bonds here for A and T. And then likewise, over here, this is the guanine and cytosine, and this is 3 hydrogen bonds. So match up between 2 DNA bases, guanine and cytosine, showing hydrogen bonds, dashed lines holding them together. So there's 3 there. And then also I added the uracil. Here's from this website. It's not from Wikipedia. Let's see the links from chegg.com. So this is uracil and adenine base pairing in RNA. So there's the 2 hydrogen bonds. There's uracil, and this is adenine. So uracil, nucleobase is adenine, nucleobase connecting together. And then this, remember, the inside is the sugar. Far end is the R group is the sugar, and when you for nucleic acids, or nucleotides and so on. So that's pretty fascinating stuff. All right, so let's continue further, and this time look at nucleic acids. And I believe that's the proper pronunciation. I've been calling it nucleic acids, but I think the proper pronunciation is nucleic acids. Yes, and nucleic acids. Yeah, so they are biopolymers or large biomolecules essential to all known forms of life. They are composed of nucleotides, which are the monomers made up of 3 components: a 5-carbon sugar, a phosphate group, and a nitrogenous base, as I illustrated earlier. And here's the bases there. The two main classes of nucleic acids are deoxyribonucleic acid, or DNA, and ribonucleic acid, RNA. If the sugar is ribose, the polymer is RNA. If the sugar is ribose, it is the ribose derivative, deoxyribose, the polymer is DNA. Yeah, which is, again, this is just a ribose that lost an oxygen. All right, let's continue further. So nucleic acids are naturally occurring chemical compounds that serve as a primary information-carrying molecules in cells and make up the genetic material. Let's continue further. So here is illustration of comparison between RNA and DNA. So here is how it looks like. So here's a strand. This is the this is actually the phosphate backbone, or the carbon phosphate backbone right here, all the way across, and then the insides are the each of the nucleobases that extend out, which connect and make base pairs. When they connect and make 2 strands, or they fold in on itself, and so on. Yeah, so nucleic acids, RNA left and DNA to the right, and there is the cytosine, guanine, adenine, uracil of the RNA, nucleobases of RNA, so CGAU, and there's the these ones are the same ones here. The only difference is the thymine is different, the T. So the uracil is replaced with thymine here, T, and this one is a pyrimidine, and this is a purine, and so on, because it's the fused ring, and these are simple rings. It's very interesting here. Yeah, and here, so helix of sugar phosphates. Yeah, so this is the sugar and phosphate make up the backbone of these nucleic acids. So let's go further. Let's look into deoxyribonucleic acid, or DNA, and this is a molecule composed of 2 polynucleotide chains, like this one here, so 2 of those chains, that coil around each other to form a double helix carrying genetic instructions for the development, functioning, growth, and reproduction of all known organisms and many viruses. DNA and ribonucleic acid, RNA, are nucleic acids, so they're all nucleic acids. So a large part of DNA, more than 98% for humans, is non-coding, meaning that these sections do not serve as patterns for protein sequences. So now that is a very interesting statement there. All right, so let's continue further. Now look at the more detailed view of the DNA double helix. So the structure of the DNA double helix, and it's a double helix, so you can see there's 2 curves there. So you'll have one like this, and then the second one comes in like that, and you'll have a double helix in 3 dimensions. And that is how it looks. And in that section, there's going to be the way it's set up is that you'll have a minor groove that's closer together of these 2 the cross sections across there, and then there's a bigger one, which is a bigger wave there of this double helix. And so the structure of the DNA double helix, the atoms in the structure are color coded by element, and the detailed structures of 2 base pairs are shown in the bottom right. So here's 2 base pairs here. This is T and A, that's thymine, and this one is adenine, or the T and A, and notice the connections here, and these are all purines on the right side. A and G are purines, and there's a G and C hydrogen bond or base pair connection there. So this is the base pair, this is the hydrogen bond over here, and this is a pyrimidine. These are pyrimidines because of the simple ring, and this is a double fused ring, the purines. And this is a cytosine. Yeah, and that's the C, and the G is the guanine. Yes, the guanine, and yeah, and there's the phosphorus. You can see the phosphorus across there. So there's phosphorus right here, and there's the carbon sugar should be here. And there's the phosphorus and the phosphate group across there, and these make up the backbone. Then in between is the base pairs, the connection. So it's very, very fascinating stuff across. Alright, so now let's look at just a simple organism right here, just to illustrate where the DNA is found. So location of eukaryotic, which are organisms whose cells have a nucleus enclosed with a nuclear envelope, and it has so this is location of eukaryotic nuclear DNA, and where nuclear DNA is DNA contained within each cell nucleus, or and DNA, so for nuclear DNA. Within the chromosomes, which are long DNA molecules, let's write, with part or all of the genetic material of an organism. Actually, I'll make it not plural here. So, long DNA molecule with part or all of the genetic material in it, and there's how the chromosome looks like. So here's, let's say you have a cell inside a eukaryotic organism. So say take a cell inside there, and then you look at it in here. It could be even a unicellular cellular organism, which has just one giant cell. And then inside there's a nucleus right here with a nuclear envelope around it. And then you go zoom even further inside there. You're going to see a chromosome, and it could include some or all of the with part or all of the genetic material of an organism. And there inside there, how it's all connected, you're going to see the DNA inside there. Yes, the chromosome is pretty much just a whole bunch of DNA all together there. So let's continue further. Now let's look at ribonucleic acid, or RNA. It is a polymeric molecule. It just means you can stack up monomers and to make a longer chain. Molecule essential in various biological roles in coding, decoding, regulation, and expression of genes. Like DNA, RNA is assembled as a chain of nucleotides, but unlike DNA, RNA is found in nature as a single strand as opposed to double strand. As opposed to double strand, you'll have a single strand folded onto itself rather than a pair of double strand. Than a paired double strand. This one's two paired double strand, but this one would be folded onto itself like that. So let's scroll over here. Let's make this a bit smaller. So a hairpin loop. So a hairpin loop like that. From a pre-mRNA, or before messenger RNA. So type of RNA, we'll get to this later in the video. So highlighted are the nucleobases in green. So there's nucleobases, and they fold onto themselves and connect with hydrogen bonds. And the ribophosphate backbone, so in blue, this ribophosphate backbone. There's the 5-carbon sugar, and then there's the phosphate all connected. Forming the backbone. In blue. This is a single strand of RNA that folds back onto itself. So fascinating stuff. Alright, so a nucleic acid sequence is a succession of bases signified by a series of a set of 5 different letters that indicate the order of nucleotides within a DNA, which uses GACT, or guanine, and then adenine, and then cytosine, and thymine. And let's just scroll up to double check the wording. It takes a while to put this in your brain. So this is the DNA one. Is cytosine, guanine, adenine, thymine. So there's an M there. Everything else then. And then for the RNA, it's the cytosine, guanine, adenine, and then uracil. Right there. So uracil, the U. So in other words, it's going to be right here. Or RNA GACU molecule. And then, so by convention, sequences are usually presented from the 5' end to the 3' end. And this is how they're numbered here. So you have, if the nucleobase is on this side, this is going to be 1. So 1, 2, 3, 4, 5. and then there is the phosphate backbone, or the sugar phosphate backbone, when you include these two together. So this is a furanose or sugar ring molecule with carbon atoms labeled using standard notation. The 5' is upstream, the 3' is downstream. So yes, so you'll consider it from the nucleobases. This is going upstream there, or actually the 3 to 5. So this is going to be upstream, the terminology, and then downstream is from the 5. Actually, no, 3 is downstream. No, this is correct. So up to the 5, that is upstream. So the 5 is upstream, and 3 is you're going downward to the 3. 3 is downstream. That's so fascinating. All right, so that's just labeling and terminology there. Yeah, so let's look at this further. So DNA and RNA are synthesized in the 5 to 3 direction. So they're created in this direction. So that's how they're formed when they're being made. It goes from the 5 to the 3 direction. Nucleic acids can only be synthesized in vivo or on whole or living organisms in the 5 to 3 direction, as the polymerases that assemble various types of new strands generally rely on the energy produced by breaking nucleoside triphosphate bonds to attach nucleoside monophosphates to the 3' hydroxyl OH group via a phosphodiester bond. Yeah, that's I have to Google the pronunciation. All right, so let's unpack that again. So nucleic acids can only be synthesized or created in vivo or on whole and living organisms in the 5 to 3 direction as the polymerases. Polymerases are an enzyme that synthesizes long chains of polymers or nucleic acids. Again, enzymes speeds up reaction. And enzymes are proteins that act as biological catalysts or biocatalysts, and catalysts accelerate chemical reactions. Yeah, so they use these polymerases, these enzymes, but these rely on the energy produced by breaking nucleoside triphosphate bonds to attach nucleoside monophosphates to the 3' hydroxyl group. And this is via a phosphodiester bond. So basically it needs to get other triphosphates, break them up and put them in here, and they get that energy, or these polymerases get that energy from that breaking, and then which they can add to this 5. So they'll add to the 5, 4, 3, and then so we'll add from this direction onwards. So it needs those bonds first. A phosphodiester bond occurs when exactly 2 of the hydroxyl groups, and the hydroxyl is just OH, like that, so dash OH, groups in phosphoric acid react with hydroxyl groups on other molecules to form 2 ester bonds. And just to recap, you can search ester right here in the in this Word doc. So that's why I highly recommend downloading it so you can go along with it very quickly, move back and forth as you wish. So you click all the way at the beginning. So an ester is a chemical compound derived from an acid, organic or inorganic, in which at least one dash OH hydroxyl group is replaced by an O dash alkyl or alkoxy group. And this alkoxy group is an alkyl group singularly bonded to oxygen. Thus you have R dash O. So basically you're replacing an H with this dash R. R is a generic placeholder for other molecules. And an alkyl, just for being more specific, is an alkane missing one hydrogen. And an alkane is this an acyclic saturated hydrocarbon. And let's just go back to our phosphodiester bond. And all the way over here, phosphodiester bond. So a phosphodiester bond, again, occurs when you have 2 of these hydroxyl groups in phosphoric acid react with hydroxyl groups in other molecules to form 2 ester bonds. So in this one here, it had OH on this side. Over here, OH. So you had OH here, you had OH here. Actually, there's already an O there. So an H. Then you would replace the H. Yeah, so we replace this with an O-R there. So O- connecting over here. Yeah, so over here, instead of OH, you'll have O-R there. And likewise on the bottom here, so you have 2 of them. So you have the OH becomes OR. Like that on the phosphoric acid. Now you're left with PO4 3-. And then connecting to the sugars over there. So a diagram of phosphodiester bonds, PO4 3- or 3- between 3 nucleotides. Yeah, so here you have 3 nucleotides. You have the phosphate, you have the sugar, you have the base. It's the thymine. And then you have this one, phosphate, sugar, and then you have the base, which is an adenine. And then you also have another adenine over here. And yeah, this is a phosphate, sugar, adenine. Notice how there's connected 3 nucleotides. So the 5' end has a 5 carbon attached to a phosphate. So there's the 5' end connected to a phosphate. So this 5' end is connected to a phosphate. The 5' end carbon. Yeah, so this carbon connected to it, attached to phosphate, and the other 3' end has a 3' carbon attached to a hydroxyl group. And then so over here there's the hydroxyl group over there. But yeah, this is actually a bit misleading. I copied this from Wikipedia. I'm just gonna have to, I think I'm gonna have to erase this because this 5' end, well, it depends on where you're looking at. This 5' end is connected to a phosphate, but this 3' end is connected to a phosphate. So then, and this 5' end is connected to OH, hydroxyl. So yes, I'm just gonna erase this right here. So yes, apologies for that. Yes, that was copied from Wikipedia, so I think it's more accurate. Just leave it like this. Alright, let's continue further to illustrate the different types of phosphates right here, or the nucleoside and the nucleoside phosphates. So here's structural elements of 3 nucleotides where 1, 2, or 3 phosphates are attached to the nucleoside in yellow, in yellow, blue, and green. Yeah, at center. So let's just look at this. First, the nucleotide termed as a nucleoside monophosphate is formed by adding a phosphate in red. So there's the red. There is there's the nucleoside right here. Nucleoside includes a 5 carbon sugar and the base, nucleobase. Nucleoside is just the base plus this. The nucleotide includes the phosphate. So if you have 1 phosphate, so here's the nucleoside there. There's a yellow for the sugar. Then there's the green for the glycosidic bond connecting to the base over there, and the base in the nucleobase, and the base in blue is any of the purines, the A, G, or the pyrimidines, which is a CUT, like that. And uracils for RNA, T is for DNA. And then so let's continue for this. First, the nucleotide termed as a nucleoside monophosphate is formed by adding a phosphate in red over there. Second, if you go even further, attach a second one, adding a second phosphate group forms a nucleoside diphosphate, so the 2 phosphates. And third, adding a third phosphate results in a nucleoside triphosphate, or the tri there. So that's plus the nitrogenous base is indicated by base. Yeah, so this base right there, nitrogenous base there. Yeah, it's in blue here, and this is the nitrogenous base. Nucleobase is indicated by quote base and glycosidic bond, or the sugar bond. All 5 primary or canonical bases, the purines and pyrimidines, are sketched at the right in blue over there. So so there's different kinds of phosphate. If you have multiple, you're going to have a nucleoside, a diphosphate or triphosphate, etc. Let's continue further. So a glycosidic bond, or glycosidic bond with different spelling right here. I mean, actually, no, that's the same spelling. It's glycosidic linkage. So glycosidic bond or glycosidic linkage is a type of covalent bond that joins a carbohydrate, in this case sugar, molecule to another group, which may or may not be another carbohydrate. So in the case here, you're connecting you're connecting a sugar to another group. So in this one right here, you're connecting a sugar to a nitrogenous base over there, or a nucleobase. Yeah, so let's go over further and let's read this part here. So this figure. So in the DNA segment shown, the 5' to 3' directions are down the left strand and up the right. So it depends on how you're reading it. So when you're reading it, you always go from the 5 to the 3. So if you're going to write it out, you would start over from this side. Again, it depends on where the 5' end is. In this case, that's the 5' end. And there's the 3. So notice how it goes that dashed line across there, and it goes down. This one goes up from here. This is the 5' end over here, which has a dashed line, and there's a direct line to the O group, and goes all the way up across. So if you're reading it, you would go, in this case, this is A. If you're reading it from the left side, you have to read it for, like, this one's going to be A, and then this next one, one's a C. So it's A, C, and this over here is a T. This one is a G. And then this next one is going to be, this is G, just for interesting, just to see how it lines up. So this is a C, then this green one here is A. The next one here is G, and then there's T. So that's very fascinating stuff. All right, let's just erase this and continue further. All right, so the next segment here. So yeah, so you're going to be reading it from the 5N to the 3N. So you have GC, GC, UGU, GU, CGA. This one is again for this has uracil, the U, that's for RNA. So the following DNA sequences illustrate pair double-stranded patterns. By convention, the top strand is written 5 end to the 3 end. Thus, the bottom strand is written 3 to the end to the 5 end. So so we always have the top one starting from the 5. So whenever we see a sequence like this is 5 end all the way to the 3. So you have A, T, C, G, etc. And it just keeps going all the way across there. All right, let's look further. And now the corresponding RNA sequence in which uracil substituted for thymine in the RNA strand. So I believe this one, they just took all the T's out, replaced it with the U. And and it's supposed to be anti-parallel. So when you have a long sequence, you should have it all line up here. So this is AUC, GAU. We'll see where it starts to repeat here. So this is AUC, GAU, and there's this one over there. Yeah, fascinating stuff. And then eventually, you should have it all line up anti-parallel, or at least in this DNA one. Anyways, continue further. Now let's look at ATP, or adenosine triphosphate. It is an organic compound and hydrotrope that provides energy to drive many processes in living cells, such as muscle contraction, nerve impulse propagation, condensate dissolution, and condensate is a class of non-membrane organelles inside cells, and organelles are just organs, or it's like the cell's organs. Similar to the body's similar to the body, human body has organs. Cells have organelles. Anyways, and going further, and also chemical synthesis. Found in all known forms of life, ATP is often referred to as the molecular unit of currency of intracellular or inside cells energy transfer. When consumed in metabolic processes, which are basically life-sustaining chemical reactions, it converts either to adenosine. You had to again Google translate. Google pronounce this word. So adenosine in adenosine diphosphate. So it goes from adenosine triphosphate (ATP), and then when it's consumed in these metabolical processes, it converts either to adenosine diphosphate, so 2 phosphates instead of 3, or to adenosine monophosphate, AMP, so just 1 phosphate. Other processes regenerate ADP so that the human body recycles its own body weight equivalent in ATP each day, which is quite amazing if you think about that. Yeah, that's about 150 pounds of ATP being recycled constantly every day. So let's go further. So ATP is also a precursor to DNA and RNA and is used as a coenzyme. And notice that it has that phosphate there, the triphosphate, and di or monophosphate. So the phosphate, it's a precursor to the DNA and RNA. From the perspective of biochemistry, ATP is classified as a nucleoside triphosphate, which indicates that it consists of 3 components: a nitrogenous base, this has a nucleoside. And remember, nucleoside is a nucleobase plus a 5-carbon sugar. In this case, as a nitrogenous base, and it's going to be adenine, the sugar ribose, and the triphosphate. Yeah, so almost a full nucleotide. But then this one has a triphosphate instead of just a regular phosphate. So here's a structure of adenosine triphosphate, which again, it could be more strictly called a nucleoside triphosphate right here. And so this is how it is. So notice the triphosphates there. There's 3 phosphorus right there. There's a 5-carbon sugar, and there's the nitrogenous base, which is a purine, and that is the A nucleobase, or the adenine nucleobase. let's go further, and here's the 3 different types right here. So adenosine triphosphate has 3, and then this one here has 2. Adenosine diphosphate, ADP, the other one's ATP. Then AMP is adenosine monophosphate, just a 1 there, and the chemical structure of ATP, ADP, and AMP. Alright, now let's look at a hydrotrope, and the reason is because ATP is an organic compound and hydrotrope that provides energy to drive many processes in living cells, etc. So a hydrotrope is a compound that solubilizes hydrophobic compounds in aqueous solutions by means other than micellar solubilization. And so it solubilizes basically incorporates these hydrophobic compounds, which hydrophobic is compounds that don't like water, so they don't want to mix with it. But this is essentially trying to incorporate this inside water, inside a suspension. So you're solubilizing it or incorporating that into a suspension of components that don't want to be, so they don't want to dissolve. So in micellar solubilization or solubilization is the process of incorporating the solubilizate, which is the component that undergoes solubilization, into or onto micelles or micelle. Yeah, so a micelle or micella, plural micelles, micelles or micelli, I think that's how you pronounce it, respectively, is an aggregate or supramolecular assembly of surfactant molecules. These lower the surface tension between some substances, dispersed in a liquid, forming a colloidal suspension, also known as associated colloidal system. And a colloid is a mixture in which one substance of microscopically dispersed insoluble particles are suspended throughout another substance, and insoluble, so they don't want to dissolve into it. And a supramolecular assembly is a complex of molecules held together by non-covalent bonds, such as these right here. So you have a bunch of surfactant materials here that lower the surface tension, and when you put a substance that doesn't like water, this is hydrophobic. It's a hydrophobic or insoluble material in this liquid, and then these all just form around it. So it forms a micelle, or just an aggregate of surfactant molecules around this. And now because it's around it, it can incorporate that into the liquid. And basically you have a whole suspension of this, a colloidal suspension of this. So one substance is dispersed inside, and they're suspending this other insoluble particles throughout the other substance. And here's a schematic of micellar solubilization of fatty substance in water. Because remember, fat and oils, oil doesn't want to mix with polar substances such as water. With the use of a dispersant and it's just a substance to improve separation of particles in a mixture such as a surfactant. So, and a hydrotrope is something that does a similar mechanism. It solubilizes hydrophobic compounds in aqueous solutions, but by means other than micellar solubilization. And basically the main difference between a hydrotrope, so typically hydrotropes consist of a hydrophilic part, a part that likes water, and a hydrophobic part that hates water. And that's why you can see here, this part right here, the red likes the water, connects to it. This tail one doesn't like it, so it goes away from the water into this insoluble particle. So that's what it connects to it. So it's similar to a surfactant in that it has it, but the hydrophobic part is generally too small to cause spontaneous self-aggregation, such as this one, a giant loop around that. So here's the difference between a regular surfactant, and there is the surfactant looks like this. You have the polar head and an apolar tail, and the polar one wants to connect with the water, and the apolar away from the water. So you have aggregation like this. So it aggregates automatically like that, and then in the process of the dissolution or dissolving other substances inside. So there's the this water insoluble chemical, the red, and it sort of surrounds it. So you have this micellar solubilization like this. But then a hydrotrope, it's smaller, so it's much smaller than that, and there's no aggregation. So it's just all over the place. They don't aggregate like this. But then when there is when it does aggregate, there is a water insoluble chemical, and then it surrounds around that, but not as fast and spontaneous as a surfactant. So it will look something like this. It's a gradual and much smaller setup. So it's very similar stuff.